I want to first thank the organizers for inviting me. It's always fun to give my talks to non-science folks, and that you guys always come up with cool questions that I can't predict, so it's always fun to have kind of questions coming from other areas of expertise. And so reading the bio was actually really helpful because I'm going to kind of stick to that same framework of mechanics, the interplay between mechanics and evolution and ecology. And so I'm going to talk about geckos. Feel free to interrupt. I keep it as informal as possible. If you want to interrupt and tell a story about a pet gecko you had, that's fine, we can do that. And so something that a lot of biologists think about and scientists think about for centuries is why are animals shaped the way they're shaped? And so to kind of dig into this question, we have to use an interdisciplinary approach. And so we study something called ecological morphology, which is the morphology of an animal or the way an animal's shaped in an ecological context, in the context of what it does in the wild day to day. And so to study these relationships, we have to study how the animal's shaped, measure its morphology, measure its leg length, its body size, go out and measure how fast it can run, how high it can jump, how hard it can bite, and then we can go out in the wild and look at how the animal uses these performance capabilities. And so that first relationship, the relationship between morphology and performance, is the field of biomechanics. And so in biomechanics, we typically steal a lot of tools and methods and ideas from engineering. We, in essence, treat the animal like a machine and say, you know, if the muscles can make this much force, how much torque are they applying over the knee? And we answer those kind of questions, examining the animal from a mechanical point of view. And then once we understand how the animal works, we can go out in the wild and see how it uses it. And that's where the ecology and the ecological observations and the evolutionary theory comes in. So I'm going to talk about all three of these categories with geckos. And so the morphology of specifically their toe pads are super cool because there's a ton of variety and a ton of variation and a lot of cool mechanics going on. And also geckos can stick to surfaces. They can support 4,000 times their body weight if they're on glass in the lab, which is really, really cool. And then because of these sticky feet in the wild, they're able to do stuff that a lot of other animals can't do. They can live in environments that other animals can't live in. And so kind of the main reason that geckos are a good study system for this approach is that they're ecologically and morphologically diverse. And so you've got bright and shiny diurnal species that are active during the day. You've got cryptic species that are either nocturnal or diurnal that live their lives hiding in crevices and blending in. You've got a ton of variation in body size. So this is the largest living gecko species found in New Caledonia. And some of the smallest vertebrates are geckos. And you've got them living in tropics, in deserts, in temperate environments, in leaf litter, on rocks, on trees. So they're doing lots of different things in the wild, so you would expect lots of cool evolutionary adaptations to different environments. And on top of that, specifically, the toe pads are super cool. At a large scale, there's a lot of variation. And what's really neat, too, is a lot of these shapes, these toe pad types, have evolved multiple times. And so you'll have really distantly related species that have very, very similar toe pads, and you'll have closely related species that have very, very different toe pads. So there's a lot of weird patterns going on that we really don't have a good idea of why any of these patterns are present. And then if we start zooming in closer, how the toe pads actually work is really cool, and there's a whole lot of variation there as well. And so for people who don't spend their whole life studying geckos, this is how gecko toe pads work. If we start zooming in on the bottom side of the toe pad, we can see that the toe pads are actually covered in hair-like structures. It's called seta. So these are reptiles, they're not mammals, so they don't have hair the same way we have hair with roots. These are actually just extensions of the outer layer of skin. And so their skin is made out of beta keratin. Our hair and our fingernails is made out of alpha keratin, so it's a similar protein. So bird feathers, bird beaks, snake skin is all made out of beta keratin. It's all the same relatively hard protein. And so these hairs are shaped kind of like trees, for lack of a better example. So they're tall and skinny and branched at the top. And so for the main species that we look at, these hairs are 100 microns tall. Our hair is about 100 microns wide. So these hairs are as long as our hair is wide, depending on what kind of hair you have. And then, so these seta are branched, and on the tips of these branches, they have small kind of spatula-shaped structures, and they're actually called spatula. And these spatula are small enough, they're 200 nanometers wide. So these tips are actually smaller than a wavelength of light, because if you remember, wavelengths of light are 400 to 600 nanometers. And so these really, really, really small structures are small enough to interact with whatever the gecko's walking on through a really weak intermolecular force called van der Waals forces. And so to really simplify that, the fact that these structures have electrons spinning around and the tree bark or the rock or the glass has electrons spinning around means they're weakly attracted to each other. And so the fact that these structures are small enough to just be weakly attracted to whatever surface the gecko's walking on, that's the whole mechanism of the system. And so here's kind of a cool video of a group of hairs that we have isolated in the lab. We're pulling it across a piece of glass, and the glass is connected to force sensors underneath. And so naturally, the hair is going to have this recurved shape. And so when the gecko plants its foot and pulls, that's the process we're recreating here. And so the hair actually is put under tension and acts like a spring. So these tips are being attracted to the ground through van der Waals, and as long as that hair is kept under tension, it's pulling up and away on the surface, generating friction as well as adhesion, or negative normal force. So it's pulling up and along the ground. And here you can see that we actually measure friction and negative normal force or adhesion. So it's pulling this way and this way at the same time. And so what's really interesting about this system is that as long as that hair is kept under tension, it'll keep pulling. But as soon as that hair is released, as soon as the gecko pushes forward, the hair actually bounces back to its natural shape and it can release relatively with little energy. And so that's very different than how our Velcro works, how our suction cups work, how glue works. And so that's why there's so much interest in using this as a novel synthetic adhesive, because it can generate strong adhesion, it's easy to remove, and it doesn't damage the surface, and it doesn't use any secretions or glue. It's based simply on how the hairs are shaped. And so now you guys have a handle on a little bit of the morphology. We're going to talk more about the morphology and the performance. And so this is kind of ongoing work still, and so this is kind of just, hey, look at what I'm doing with geckos. And so previously, up maybe the last 15 years or so, people have mostly used mathematical modeling to understand how these hairs work. So if you simplify the hair structure into just a group of solid beams with a known length and a known density and a known diameter and a known material property, we can write down, particular equations describing how the system should work given these simplifications. And so we've got lots of people have done lots of cool engineering trying to break the system down to describe how much friction and adhesion it should generate, how much energy, mechanical work is required to yank a group of these hairs off, depending on how they're shaped, how stiff or how springy a group of these hairs should be based on how they're shaped, and why the hairs don't stick to each other based on how they're shaped. The problem is with these oversimplifications that we make, and when we compare our predictions based on what we actually observe, they don't actually work very well. Our predictions aren't very accurate. So this is a study we did looking at how much energy is required to yank a chunk of hairs off based on what direction we pull. And so this is what we predicted, this is what we observed. And so these simplified mathematical models just aren't very accurate. They aren't very good. And one of the main reasons for that is the shape of the hairs changes along this part of the toe. And so if you measured the hairs here, and so you write a mathematical model for exactly how these tiny group of hairs should work, and then you throw the whole toe on something and measure it, you're kind of not capturing all this variation in your model. And capturing all this variation is fairly hard to do in a simplified mathematical equation. And on top of that, when we start looking at different kinds of geckos, all of these hairs are shaped differently. And so these are pictures of the seta, of the hairs, all drawn to scale. And so you can see some species have really long, skinny hairs. Some species have just small, skinny hairs. Other species have really large, thick hairs. Sometimes the hairs are set up in groups of 4 in quads. Sometimes they're densely packed. Sometimes they're very thinly packed. And so the mathematical models kind of can't deal with this variation as well. And so for my postdoc, what I came up with the idea, instead of trying to simplify the system and write a mathematical equation to describe it, steal another tool from engineering and use a simulation approach. And so use micro CT scanning. And so whenever you get like a CAT scan of your shoulder, this is the same process, same mechanism, but it's much, much smaller. So we're able to see nanoscale and microscale structures. And so this is often used so this is a picture of a fossilized spider from amber that was imaged. And so once we have this 3D reconstruction based on X-ray, then we can actually use a tool from engineering called Finite Element Analysis, which is a simulation tool where you take a complex 3D structure, usually something an engineer designed in a CAD software like a piston or a gear or some sort of clip. If you tell the computer exactly how the object is shaped, what kind of materials it's shaped out of, and start applying force to it, it can predict how that force propagates through your complex shaped object and predict how it'll deform and bend, like what this cool YouTube video is showing. And so that's the plan to do that with gecko hairs. I know how the hairs are shaped from CT scanning. I know what kind of material properties they have because they're a protein and other people have studied that. And then I'll be able to do these simulations to actually simulate how the hairs bend and move instead of trying to write down equations to simplify it. And so far right now, I'm only done with this part. So that's the part I'm going to talk about. And so this is a 3D reconstruction of a group of gecko hairs. And so this was done from a micro CT scanner at UC San Francisco in the dentistry department, actually. The dentistry departments tend to have the micro CT scanners that are high enough resolution to be able to see these really tiny hair-like structures. And even these high-resolution CT scanners aren't quite high enough resolution for what I want to do. You can see the tips. I'm getting the overall shape right, but I'm not really getting any of this tip structure. It's all just kind of globby stuff because the resolution's just not there. And so the next step, what I'm currently working on now, is working with folks at Argonne National Labs outside of Chicago. So Argonne National Labs is a Department of Energy research site. It's a synchrotron, it's a particle accelerator. And so with this particle accelerator, they have X-ray beams that come off that are very, very high resolution. They're down to 80 nanometers. So they should be able to image the things I want to see. So I've been out there a couple times, and these are super raw X-ray images from some of the images we've gotten. So these images are still being processed by the folks there so that I can actually build my 3D reconstructions from them. But if you squint, you can kind of see this is a group of hairs kind of shaped like this. You can see that this is kind of a group of hairs shaped kind of like this. And so it looks like once these images are done being processed, we will be able to see the tip structures, which will be really cool. And that'll help the 3D reconstructions. So that's kind of an ongoing thing. And it's just cool to tell people that I get to put geckos in a synchrotron. So now you guys have a handle on kind of what I'm working on between the morphology and performance. Now I just want to talk about performance in general. So a lot of times what people do to study how sticky a gecko is you plop it down on a force sensor and drag it along a surface and measure how much friction it makes. Another way to do this is to measure something called angle of toe detachment, which is the test that I've been using. And so this is a live, non-sedated, slightly angry gecko hanging up by just a single back toe on a very, very clean piece of glass. And as this glass is slowly inverted, I can measure what angle he falls off at. So this is our rotating stage, and then this is a force sensor up here to tell me when the weight changes, so I can calculate exactly when he detaches.
So is it different if you use a different toe?
Probably, but
We don't know yet.
we don't know yet. We try not to talk about variation because every possible variable varies. And so we try to just use either the longest toe or the center toe just to try to stay as consistent as possible. And so you're thinking, well, this probably has to do with how much the animal weighs, right? Because if this was a piece of Scotch tape, the more weight you put on it, the earlier it'll peel off, right? That's not true for this system. And so a professor I worked with at Portland actually strapped little backpacks to these guys, artificially increased their weight, and they still always fall off at the same angle. And the reason for that is if we think about the bottom of the toe pad has these tiny hairs on it, right? So when the glass is vertical, these hairs are pulling nearly parallel to the glass, generating a lot of friction, but they're not generating any adhesion, any negative normal force, because they don't have to. As that glass becomes more inverted, I'm going to try to stand over here. As the glass becomes more inverted, this angle between the shaft of the hair and the glass is increasing. Now the hair needs to pull more indirectly, more perpendicular to the glass. And so eventually the hair's bending, and the hair reaches a point when it wants to snap back to its original shape, and the whole system just fails. And that's when the animal falls off, is when the hair reaches its kind of max hyperextension, if you think about it like that. And so that's,
Rotate the other direction, it falls off really fast.
if you rotate the other direction, then the toe's actually gripping on top, and he'll stay as long as he wants.
Stay as long as you want.
Yeah, because then now you're gripping up and over the edge, you know what I mean? Yeah, because usually they're hanging here on the bottom edge. And so if the glass started going this way, they would just grip. And so this maximum angle is kind of getting at this maximum ratio of adhesion to friction that we can measure with a whole group of hairs. And so it's kind of a different way to measure performance, but it's kind of the way I've been doing it. And so I don't really expect you to be able to read any of this, but I've done that same test for 50 some odd different species of geckos and anoles. So anoles are a completely different group of lizards that are found in the Caribbean and South America that have really, really similar toe pads that they evolved them independently. And so this is kind of a simplified family tree of all lizards. And so you've got geckos, which are this big group down here. There's 7 families of geckos. Carpodactylin geckos don't have any toe pads, so I don't have any data for them. Pygopodiae is a family of geckos that don't have any legs. They're convergent with snakes. And these 3 guys are only found in Australia. So it's a cool, neat thing that there's a whole legless group of geckos, but they don't have any toe pads. Diplodactylin geckos, which are, again, only found in Australia. I've measured a handful of species for them. Eubiphidae, so if anybody's had a pet leopard gecko, that's where they'd be. They don't have any toe pads. Spherodactylin geckos, which are South American, have toe pads. I just haven't been able to find any in the pet trade or go to South America yet. So if anybody has grant money, I need to measure more spherodactylin geckos. And most of what I've measured is Geckonidae geckos, which are found pretty much everywhere else around the world, which are your tokay geckos or your house geckos if you've been in Hawaii or Florida. And then the rest of lizards are all up here. Your skinks, your snakes, your chameleons, your iguanas, your fence lizards, your horned lizards, your collared lizards, and then you've got anoles. And so these colors represent kind of what angle they could stick to. So the warmer colors means that they could stick a lot longer, which means they're generating more adhesion. The cooler colors means they fall off really early, which means they're good at generating friction, but not negative normal force, not adhesion. Yeah? Right around 40. So I've got a 40, and 2 39s. So if you think about it, at 45, you're able to generate equal parts friction and adhesion, which is fairly remarkable. And I feel like that's probably an upper limit. I don't really have a mechanical reason for why yet. But we do see a couple species get near 40 or at 40, which is nearly a 1-to-1 ratio, which is pretty impressive. But most of the anoles are down at 15 or 20 degrees, which is more like a 3-to-1 or even 4-to-1 ratio of friction to adhesion, if that makes sense. And so looking at this data across all these species, we can see anoles tend to be lower, 15 or 20 degrees. Diplodactylin geckos are kind of in the middle, and Geckonidae geckos seem to be the overall kind of a stickier group of geckos. And so we were really curious. We know anoles evolved toe pads independently from geckos, and so based on the family tree, which is derived using DNA that I didn't do, and all of my performance data, we were wondering if we could predict kind of how performance has changed through time, fitting different using fancy statistics. And so I was working with another postdoc, Joseph, in Luke Harmon's lab. And so that was our question: do these independent origins of adhesive toe pads show different evolutionary patterns? So based on the tree and the distribution of data that I have. And so this is something that we're currently writing up, and so this is why this graph looks kind of goofy. But pretty much what we found was, so anoles are a lot younger than geckos, and we're assuming that based on fossils that when anoles evolved and the fact that when they first evolved they didn't have toe pads, and then when they first evolved toe pads they didn't work very well. That's kind of our working hypothesis. And the same thing for geckos. When they first evolved they had a performance of 0, and then that quickly changed to become what they currently are now. And so fitting all of this data into a Bayesian statistics framework, pretty much what we found out, or what the data suggests, is that when anoles evolved toe pads, they changed very quickly. They the performance capabilities changed very quickly over time, and then they leveled out and stayed very constant through time, which is a different pattern than what we see for geckos. So the data for geckos suggests that geckos evolved toe pads and kept just slowly changing through time. And the reason we can kind of tell these patterns is that if a trait just keeps slowly changing through time, you would expect the species that are more closely related to each other to be fairly similar still. But if you change very quickly and then stay constant, while kind of continuing to change within this really narrow band, closely related things may not be as similar as distantly related things if you're kind of keep dancing around in this really small band of area of performance space. And so that's kind of how we can tell these different patterns apart, is by comparing closely related species and distantly related species, given the family tree and the performance capabilities. So this is kind of a cool result that's suggesting that maybe anoles and geckos evolved under different patterns. So we've talked about morphology, we've talked about morphology and performance, we've talked about performance. Now we're going to talk about ecology. And so as part of my PhD, I got funding from National Geographic to go to Australia and catch geckos. And so when I was planning this trip, I had a really tough time understanding how big Australia is. And so here's a map of Idaho drawn to scale to Queensland. Yeah, and so we started, I was there for 5 weeks. We started in a town called Townsville, working with a professor, Lynn Schwarzkopf from James Cook University. And we drove all the way up to the tip of the Cape Peninsula, which is like cool, tropical. We're right near Papua New Guinea and Indonesia when we're up here. And so this is tropical, warm, wet environment. We did a loop, came back to Townsville, and then we went out into western Queensland, which reminded me almost strikingly similar to Nevada, kind of middle of nowhere desert scrub. And so I got to design some of my own equipment, so I couldn't take the lab toe angle setup that we had, so I got to design my own. So I had the guys here at facilities TIG weld me up a frame, and I threw some force sensors and stepper motors in it and wrote a computer program to run everything and measure everything, and it's got accelerometers on the back. So that was cool. I got to pretend to be an engineer for a little bit. And then, so we went into this project with kind of some questions based on other lizard studies. Our first kind of very generic question is that different kinds of geckos are probably going to be doing different things in the wild. And then specifically based on studies of anoles, we know that anole lizards that have short legs tend to live on skinny branches. So we were thinking, well, that's probably true for geckos too. And then studies of skinks have looked at skink species that live on rocks and have longer legs. So maybe that's true for geckos too. So we went out, we were able to catch 70 different animals from 13 different species. So we measured what kind of thing they were living on, how high they were off the ground, what diameter, if they were on a tree or a bush, and what angle that perch was on, what angle of the surface was. And then I measured their leg lengths and how sticky their feet are. And so kind of what we found at first was, yeah, different species do use different types of environments. So you've got vegetation, rock, and being on the ground. So some species were only terrestrial, only arboreal. So we only found them on trees or bushes. Other species we'd find sometimes on trees, sometimes on rocks. Other species we found only on rocks. And then some species we even found on the ground fairly often. So this kind of tells us, yeah, different species are doing different things. I know it looks kind of crazy, but we're going to walk through it. So this is how sticky their feet are compared with how high we were observing them off the ground. How sticky their feet are compared to what the diameter, if they were on vegetation, what the diameter of the branch was, or if they were on a rock, how big the rock is. And then how sticky their feet are compared to the perch angle that they were on. So given this whole set of data, we can actually kind of start to tell a story of what part of the environment they were living in. And so this, where am I at? This is a Gahaira gecko. This is one of the species that we were looking at, and they're the black dots. I almost need a laser pointer. So these guys, we could see, had sticky feet and were typically a meter or so off the ground. And they were on fairly thick branches. And these branches were always nearly vertical at 90 degrees. So in essence, they were always on tree trunks, right? Thick, vertical, about a meter high. The next species that we observed from the Strophaurus genus are the gray dots. So they were always very low, very near the ground, on very skinny branches, and these branches were always nearly horizontal. So they were in bushes, is where we always found Stropharis. Pseudothecodactylus, which is the blue, we found super high up in the trees, very sticky feet, and very high up in the trees, 4 meters, or 10 feet or so, on a variety of diameters and a variety of angles. So he was living in the canopy, kind of just doing whatever he wanted to do, being the top predator, big angry gecko in the canopy of trees. And so some of the Odura species we saw, very high off the ground, but always on rocks. So we'd see them on like rock faces. And so kind of by measuring all this data, we can actually kind of start to tell a story of quantify the habitat that they're living in. What's also cool is we were doing some other statistics and we found a pattern that we weren't really looking for, which is kind of cool. When we compare how sticky their feet are versus their leg length, we find out that species that have longer fingers, larger hands, have stickier feet, which is we weren't really looking for or expecting that relationship. But now we have a whole new kind of mechanical hypothesis to go down. Why would having stickier feet work better with bigger toes? That's a whole nother question that we weren't even thinking about that now we have data for, which is kind of cool. And so in the end, we found that species do use their environments differently. We found that, I didn't show you this graph because it's not very pretty, it's just a big blob of dots. But species with longer limbs were using narrower perches. So this is the exact opposite. So what's really weird, oops, wait, wrong way. These guys that use these really narrow perches have really long legs. They're kind of long, spindly guys, which is the exact opposite from what we were expecting from anoles. And so the reason we think anoles have short legs is to trade balance for speed. So if you're on a flat surface, you can have long legs and run quickly and not need to worry about falling off. But if you're on a short branch or a skinny branch, you need short legs and you sacrifice your speed for balance. Are geckos not doing that? Do geckos not need to trade balance for speed somehow? How can they have long legs and still, control balance and speed? And so there's something different going on possibly with geckos, and possibly their toe pads have an influence on balance and speed maybe. We also found that the species that live on rocks do have longer legs, and we found that species with this stickier feet have longer feet, which was a cool pattern that we can kind of develop new hypotheses from to keep going into the future. And so we've talked about morphology, we've talked about all these categories. And so this is an interdisciplinary talk, so at the end, I'm kind of obligated to include that geckos are a cool interdisciplinary system. Hopefully I've convinced you of that. And on top of all the stuff I've talked about with the evolution and ecology and biomechanics, there's a whole nother world of biomimicry. And so there's a lot of engineers, material scientists that are interested in learning how geckos work so that we can copy them using synthetic adhesives, synthetic materials to build Spider-Man gloves and gecko robots. And there's a lab in Massachusetts that's developed something called Geckskin, which on an 8x11 page can hold up 800 pounds on glass, which is really cool. So there's people out there developing really, really cool synthetic adhesives for lots of different purposes, kind of based on our understanding of how geckos work. And that's it. Do I have any questions? Yeah?
You said that there was varying amount of diameters of the hair on the toes. Is there a correlation between the detachment angle and the
density?
number of hairs? Or yeah, the density of...
I haven't actually looked at that. I'm not sure. There is a relationship between length and detachment angle.
The length of the hairs?
Yeah. And my gut would tell me that there's probably a correlation between length and density. Probably an inverse relationship. The longer you are, I think the less dense your hairs are. But stiffness and diameter come into a role there. Because there was a study that looked at why don't the hairs stick together? And pretty much if they're longer, and they can either be longer, stiffer, or skinnier, or less dense. So you can kind of change all three of those variables to make sure you're not too close together. And so all four of those variables work together. And so I don't know how that would work with performance. But that's something with the X-ray simulations we could do that. And then what'll be really cool is we'll be able to take our 3D reconstructions, our x-rays, and manipulate them, artificially make them more dense, try to run them and see what happens. So we can even test morphologies that don't really exist to understand where the system breaks, which will be really cool. Yeah?
If you let them hang on with their whole foot versus just one toe, is the angle similar?
It doesn't work. And so what happens, if you look at their toes, come on, usually their toes are kind of in this star pattern. And so what happens if you give them the whole foot, their toes pull in different directions, and so they can ride it fully inverted. Because now you've got toes acting almost kind of like a suction cup. Everything's pulling towards the center.
So it does work, it works better.
Well, yeah, that's the same way they can run across the ceiling in your hotel room in Miami, right? And so, yeah, if that's why we have to limit it to one toe, because now all the hairs are pulling in one direction. When we let the hairs pull in more than one direction, they complement each other, and so they don't have a failure angle anymore. Mm-hmm. Yeah.
So you're testing them all on glass, but like are some toes better suited for different surfaces than others, or
Well, define better suited.
like stickier to the
Yeah. And so that's kind of one of the weird things about this is, so usually when you're studying wild animals and you bring them into the lab, usually they perform less well than they may in the wild because they're stressed out or they're not under the right light cycle or whatever. There's 100 different reasons why when you bring something into the lab it might not perform as well. But geckos actually do the opposite, where if you stick them on glass, they perform way, way better than they do on natural surfaces. I know where I'm going, hang on. And so actually in this study that I stole these pictures from, he looked at how he took a cross-section of sandstone, where these guys are found in Africa. And so pretty much based on how the hairs are shaped and the orientation they are in the foot, he kind of calculated conservative estimates of how much of a real-life surface like sandstone these hairs would actually touch. And he calculated they're probably only coming into contact with 9% of the surface of a real-life surface. And so at 9%, calculating how strong each hair is about, these animals are probably only barely supporting their body weight on a real-life rough surface. But when we bring them into the lab and put them on a perfectly flat surface, now all the hairs can touch and make proper contact. And so that's why we think they're overbuilt, because when we run them on glass, they are. Because the smoother the surface, the more hairs that can make contact, and the better off they do. And so, but that's a whole interesting question. There's a lot of theoretical work that's been looked at how different size oscillations should affect how the hairs make contact with the surface, how the whole toe pad makes contact with the surface, and how those really, really tiny tip structures make contact with the surface. So if you have oscillations, depending what scale, can have an impact. And so we think there's probably room for adaptation there, but it's hard to measure how rough a surface is at that scale. It's hard to measure how long these hairs are across the whole foot. So that's something else I want to look into. Mm-hmm.
In the video you had, the geckos seemed to be fairly cooperative.
Yeah, and so there's those guys that are actually I had those individuals for a year or two already, so they kind of knew. Oh, I think I went past it. They kind of knew what was coming, and so this species is Phelsuma madagascarensis, which is the same thing as the Geico gecko. So you can see he's green. He's got some orange dots here. He doesn't talk, but and so typically these guys are a really, really bright golden, bright emerald green. And so you can see here he's kind of olivey colored, and so they can change color a little bit to kind of express their personality or their attitude. So the fact that he's kind of this dark olive means he's upset and he's stressed out and he's not happy.
Is that why when he hits the bottom,
He just plops. Well, he doesn't run.
Yeah.
Because there's kind of this, you can stress them out enough to where they kind of quit fighting you. But then you have a little plateau, and then if you keep stressing them, they can actually pass out and not do so well. And with the bigger lizards, you're okay, but with the really, really small ones, they have a harder time regulating their body temp. And so when you stress them out, you have to be really careful because they can overheat.
So do they recognize you? I mean, do you have any kind of relationship with them?
So I don't think it was this guy. I think it was another one in this same species. We had 4 or 5 of a handful of species. When you'd walk in, because we feed them crickets, so you'd walk in, one of them, it was a big brave male, would run up to the top of the cage and eat crickets out of your hand. So kind of, they at least associate you with food, or this, one or the other. So you have a mixed kind of association. Yeah.
So it kind of seems like they're unwillingly like stuck to the glass.
At
Like,
in this position,
they
yeah.
can't control their own toe pads, like if they want to release themselves.
So they have a couple different ways to release. And so one way that they can do it is they can push forward to bend all those hairs back. Another one that they can do is they can hyperextend their toes. So they can actually roll their toes back almost to where the tip of their toe touches back here. And so that's another way that they can detach. And they actually do that when they're running on flat surface. They actually keep their toes hyperextended to keep their toe pads up and away to not interfere with how they're trying to run. When they're stuck in this position, all their body weight is hanging on that one toe. So they can't push up and they can't hyperextend. So they are stuck. And they know they're stuck. And so that's the reason the first couple times, like when I'm doing field work, the first couple lizards I grab off a tree and try to put them on here, they know they have 2 options. They can either fall, because they feel themselves falling. They can either wait for it or they can just spaz out and fall. And so usually they spaz out and fall the first couple times until they just kind of reach that stressed out stage where they give up fighting you, and then you can actually do the test. But otherwise they're like grabbing the pole back here because they don't have enough strength in their leg to do a complete lift of their whole body weight. So they're kind of stuck. And you just have to convince them to stay there long enough for the test to happen. Mm-hmm.
Could you speak a little bit about your approach to interdisciplinary work? Are there times when you find yourself thinking like an engineer, and other times thinking like a biologist, and other times like an ecologist?
So I think,
let's just go back
yeah. So I have, like I said, like you said, I have a degree in cellular biology, so I took a year of physics, but that's really my only formal engineering training. But for me personally, the engineering comes a lot easier. So I usually I'm learning the engineering on the fly. Like find an element analysis, I'm kind of just teaching it to myself, and I can kind of limp along with that well enough on my own. I've learned all the evolution and ecology, so it's fun to think about what one means for the other, is usually kind of the way I'm thinking about it. Or the other way too is if I'm thinking about it from, how is this animal using its toe pads in the wild? Okay, I have an idea how. Now how can I test it, or how can I build a piece of equipment? So yeah, for me, I'm always jumping back and forth between like, what does this mean versus how does this help the animal find food, or how does this work, or how can I build something to test it? Or what kind of statistics do I need to measure it, which is horrible, which is something I struggle with, so.
You mentioned that your trip to Australia was funded by National Geographic.
Mm-hmm.
Will this end up in the magazine? Or
They did a blog post about it a while back. Yeah, it was a very simple, just kind of a blurb about somebody's going to study geckos, kind of similar to like the U of I College of Science study. But it was a relatively smaller grant through them just to study kind of young investigators, and so it wasn't one of their full-blown. I suppose if I had found something really cool, maybe it could have been that way, but I kind of just found simpler patterns.
Anything else? Well, I invite you to stick around after.